Cellular principles of invasion and metastasis refer to the mechanisms by which cancer cells acquire the ability to invade surrounding tissues and spread to distant sites in the body. The process where cancer cells separate from the primary tumor and infiltrate the surrounding tissues is referred to as invasion, and the spread of cancerous tumors from one organ to another is known as metastasis. Cancer cells are attached to the epithelium by E-cadherin. E-cadherin is a transmembrane protein that interact with other E-cadherin protein. Cancer cells use colony-stimulating factor to attract macrophages and other stromal cells. Macrophages then release epithelial growth factors, which cause more CSF to be released, causing a positive feedback loop. Macrophages then release cathepsin B, a protease that cleaves the attachment of E-cadherin to the epithelium. Macrophages also release more EGF and tumor necrosis factor alpha to stop the transcription and production of E-cadherin and increase the transcription and production of N-cadherin. Stromal cells release matrix metalloproteins that degrade the basement membrane. This causes chemoattractant factors like hepatocyte growth factor to be released, acts on tumor cells, triggering RAS-like GT-PACE actin rearrangement. Cancer cells then make broad, flat lamellipodia in the direction of the HGF, forming new adhesions at the beginning and losing old ones at the end. MMP will clears the passage for the tumor cells in the extracellular matrix by cutting or dissolving the extracellular matrix component. The intravasation process happens when there is interaction between N-cadherin on the cancer cells and N-cadherin on the vessel epithelial cells by increasing the permeability of the vessels and allowing the cancer cells to enter the bloodstream. Once the cancer cells enter the circulation, it can be called as circulating tumor cells, or CTC in short. This CTC will circulate in the vessel either in single or in a cluster. This CTC need to adapt with the microenvironment and obstruction in the vessels, such as shear stress, oxidative stress, and NK cells attack. The adaptation process of the circulating tumor cells can be done by metabolic adaptation, platelet coating, and neutrophil interaction. Extravasation of the cancer cells is the movement of the cells out of the vessel into the tissue, or it can be described as the fluid leakage, such as blood or lymph fluid from a vessel into the tissue around it. The extravasation of tumor cells can occur in 3 scenarios. The first one is when the circulation tumor cells are entrapped in small capillaries. Organs with sinusoidal vessels, such as liver and bone, help in the easy penetration of the cancer cells. Lastly, transmigration of the cancer cells. So, this is what happened in the transmigration of the cancer cells. The tumor cells will attach to the integrin on the vessel epithelial cells and do the rolling action to tighten the adhesion. Once it reaches a specific site, the tumor cells will arrest and give rise to significant cytoskeletal changes. After that, the transmigration process will occur. Once the tumor cells undergo extravasation, the tumor cells will migrate near the blood vessels, and when it arrives at the favorable site, the tumor will colonize the site. The growth of the vascular network is essential for the metastatic spread of cancer tissue. Angiogenesis is the process of forming new blood vessels from pre-existing blood vessels. The function is to supply required nutrients and oxygen for the metastasis process. Angiogenic process is regulated by maintaining inhibitor-activators balance. There are 6 steps in angiogenesis. First, tumor cells will release angiogenic factors or activators. It will bind to the endothelial cells of neighboring blood vessels, causing activation of these endothelial cells. Following this activation, endothelial cells will produce enzymes that break down the basement membrane to create tiny pore. This tiny pore will undergo proliferation and migration towards the angiogenic source, which is tumors. A variety of mechanisms are used, such as adhesion molecules and enzymes, to aid in movement towards source. Next, endothelial cells of a new blood vessel will roll to form a tube-like structure, or called as a loop, between source and the existing blood vessel. Lastly, this blood vessel loop will undergo structural stabilization with the help of smooth muscle cells as it provides support to the vessel. Here are several activators and inhibitors with their role in promoting or preventing angiogenesis, respectively. In conclusion, metastasis is the ultimate stage in cancer and the major cause of death. Despite gaining more attention in research, the mechanism remains unclear and effective treatments to counteract it are restricted. However, with greater research and understanding of the metastatic process, effective therapies may be developed.